Now is that audible? Okay, now it's coming. So still it's not audible. I hope my voice is reaching. Huh. Okay. So how are you doing? Everyone fine? Namaste. Namaste, good morning. Good morning, Janvi, good morning. Atcom. Maheshwari, I'm good. How about you? Kartika, good morning. Good morning. Oh, that's really great to see everyone is in good mood, or we can say for everyone it's a good morning. Seti, I'm also good. How are you? Good morning. Okay, so it's a really good morning. Means I have seen a lot more message or a lot more chat. By seeing all those, I can say that everyone is in good state, or we can say it's a good morning. Okay, so in the last class we have seen something. Okay, everyone had your breakfast. Everyone had your breakfast. I think yes. Achha, no, why? Are you in fasting, or we can say dieting mode? Like before going to the college, we need to, or you need to lose your weight. Okay. It's too early. Okay. So we'll move to the next one. Next one is detoxing day. Yogendra. Don't detox because you have not toxified yourself. So don't detox much. Idli and vada is good. Achha, no time. Dragon still 60 kg. So increase, make it century. Okay, after my class. Mamta. Okay, good. So in the last class, we have seen the structure of what? Structure of the prokaryotic cells, or we can say it's a structure of bacteria. So if you are talking about the structure of bacteria, the outermost layer which we have seen is nothing but it's a glycocalyx. So let's go in a little bit brief about the glycocalyx. So in the first structure, and that we can consider it as what? So we can put into it is known as what? Cell envelope. So in the cell envelope, how many structures are coming? Firstly, the outermost layer is known as glycocalyx. After that, which one will come? After that, cell wall will come. That is rigid, permeable structure, means it allows everyone to pass through it. After that, the most delicate, the most vulnerable, the most beautiful structure, that we can call it as a cell membrane, or we can say it's nothing but a plasma membrane, biomembrane. So it is known for its a different name. So we'll go one by one. So the first structure that we can encounter is nothing but the glycocalyx. So can we say that glycocalyx is nothing but the outermost structure? Glycocalyx is nothing but the outermost structure, and in this glycocalyx, mostly we can say it's nothing but the polysaccharide. So mostly, what is that? It's a polysaccharide. So mostly means we can't say that only polysaccharides are present. Other than the polysaccharides, we can find something else. We can find something else. So that's why I'm writing it's a mostly polysaccharide. Okay. Gram positive, gram negative, Janvi, we'll go the structures in brief, so accordingly we'll deal. So it's a polysaccharides. After that we'll move to the next one. Can we say it's a outermost structure? Obviously we can say it's a outermost structure, but it can be of two types. It can be of two types. So one, we can say it's a slime layer. Slime layer, the thin layer, the transparent layer, that is known as slime layer. That is known as slime layer. Dragon, is it made from Golgi and E? Obviously, there is no Golgi body. There is no endoplasmic reticulum present in the bacteria, because bacteria is what? It's a prokaryotes. Whenever you are saying prokaryotes means obviously there should not be any cell or that is membrane bound cell organelle. Okay. And what is polysaccharide? So polysaccharide, poly means what? Poly means it's a more than one. And saccharide means what? Sugar. Or we can say it's a glucose. So glucose is nothing but the monosaccharides. Glucose is monosaccharide. So let's say you are having more than one monosaccharide. That is a 1, 2, 3, 4, 5. 5 monosaccharides are there. Or we can say 5,000 monosaccharides are there. So that combination of 5,000 monosaccharides is known as what? Polysaccharides. Is polysaccharides. Yes, long chain of carbohydrate. That is also true. So overall we can say saccharide means sugar, means the complex form of a sugar is known as polysaccharide. And obviously it's the outermost layer, or we can say it's outermost structure. Sucrose, Tulsi, sucrose is not polysaccharide, sucrose is disaccharide, because it is having two monosaccharides, that is a glucose and fructose. Glucose and fructose. So that's why we can't say that it's a polysaccharide, it's a disaccharides. And glucose. So glucose is the monosaccharide. Why it is monosaccharide? Because we can't dissolve, we can't further simplify. We can't further simplify. Chapter name, Hemsikha, the chapter name is cell biology. Cell biology. Glycogen. Glycogen is a polysaccharides. Okay, so we'll move to the next. So if you are talking about the types, the first one is known as slime. First one is known as slime layer. Next one is if we are adding some point to this, slime layer. So slime layer is thin and slimy. And if it is a thin and slimy means water content is much. In this case, water content is more. Water content is more here. So that's why it's a thin and slimy layer. Okay. Next one is it mostly consists of, so here it's a mostly consists of what? So mostly consists of polysaccharide. Mostly consists of polysaccharides. And obviously it helps in protection. It helps in protection, or we can say attachment to the substrate. Let's say something is sticky. So can we say that it can directly stick to this part, or we can say it helps in sticking to some other surface. But obviously the main component is fimbriae. Which helps in attachment to the substrate. No doubt it is also helping. So protection. So it's a protection. Achha, whenever you are going outside, and outside, let's say the temperature is 45 or 46 degrees centigrade, 45 to 46 Celsius. What do you to protect yourself? What do you do to protect yourself? So I think one is sweating. Sweating is one part. But if the sun directly falls on your skin, or we can say your body, so sometimes it can damage your skin. So for that, what to do? Yeah, white dot, sunscreen, Usarani. Yes, you can take umbrella. Uh, and other one is also like sunscreen. So sunscreen is good. So whenever the sunlight falls on this surface, most of the part is reflected, or we can say it is well protected. So for that, we are using the moisturizer, or we can say sunscreen. So slime layer is also sunscreen for what? SPF 50, yeah. Risvita. So here it's a slime layer. Sunglasses. So with the help of sunglasses, you can protect your eyes, not the skin. So obviously, can you imagine a bacteria with the sunglass? Can you imagine a bacteria with the sunglass? So obviously, it's a very difficult, or we can say too weird to imagine this type of things. Okay. So that's why slime layer is nothing but it's a SPF, or we can say sunscreen, or we can say it's a protective layer like a moisturizer. So this is the first part of slime layer. Okay. So next one is, after that, we'll move to the next part. So second thing is nothing but a capsule. So can we say that slime layer and capsule is a type of glycocalyx? Is a type of glycocalyx. So here we can say capsule. If you are comparing the slime layer with the capsule, then we can make few differences. So what are the difference? First one is it's a thick, not transparent. It's a thick. So here it's nothing but a thick layer. In this case, you can find few amino acids along with the polysaccharides. So along with the polysaccharide, you can find few amino acid, not all. Few amino acid. Okay. After that, again, it's a function is protection. Let's say one bacteria enters in your body, and your immune system is too strong. Your immune system is too strong. So what your body will do against the bacteria? So obviously your body will start fighting with the bacteria. Start fighting with the bacteria. So who will win? Who will win the match? It depends. It depends on the condition. Like whoever play well will win the match. Will win the match. So obviously, if your immune system is not so good, you will be getting some kind of disease or some kind of infection. But if your immune system is too good, then obviously you can kill, you can defeat what? You can defeat the bacteria. But while fighting, the bacteria will develop another type of layer that is known as capsule. That is known as capsule. And this capsule will fight against your immune system. Will fight against your immune system. RCV will win. Okay. That depends, like whoever is playing good will win the match. Okay. Anyway, it's not... don't confuse with RCV and RBC. So RBC is a good thing. RCB is also, but RCB is good for the RCB. But for all, RBC is good. So here it's a capsule. And again, the main function is, again, we can say it's a protection. Or here we can say protecting the bacteria, from adverse condition. So from adverse condition. What is adverse condition? Okay, so sometime you can find yourself while getting infected. While getting infected, your body temperature will become it's a high. Body temperature will increase, or we can say you can call it as a fever. So fever is good or bad? Can anyone tell me fever is good or bad? No, actually fever is good, or we can say it's like a physiological system. So whenever our body encounters an enemy from outside, so obviously our body will try to kill that, or we can say our body will make conditions unfavorable to that coming person. So by increasing the temperature is also the way of killing that unknown person, or we can say it's a devil person, or we can say enemy that is coming from outside. Okay. And anyway, like fever is good, but persistent fever is not good. Persistent fever is not good. Like for one day it's good, but it's like years and we can say months, it's not good. Okay, so that will just weak your body itself. Okay, so here we can say it's protecting the bacteria from adverse condition. Adverse condition can be anything, like here we can say pH, temperature, or we can say any other conditions that will make it adverse, or we can say not able to survive. So this is the one part, or we can say we have discussed one thing that comes under the cell envelope. So what's the next thing? So next thing, can we call it as a cell wall? So cell wall is dead or rigid, dead or living structure? So it's nothing but the dead structure. And if it is a dead, then do they have any sensitivity, any emotions, any feelings? So obviously not. It's a dead. It's dead. It won't act differently to the different things. It won't act differently to the different things. So cell wall is, we can say, just inside that, just inside the glycocalyx. Next point we can add is it's a dead one. Next one is it's permeable or impermeable. It is permeable. It is not human being. Obviously, human being. It's not human being. It's a bacteria. But still, bacteria is also having the feeling and emotions, right? So just inside the glycocalyx, and it's a dead. If it is a dead, means it is not having the power to discriminate the things, to separate the things, So that's why we can say it's a permeable. It is permeable. So cell wall is permeable. After that, what else we can add? After that, we can say if it is a components. If you are talking about the components, so bacterial cell wall is known as peptidoglycan. Bacterial cell wall is, or we can say component of bacterial cell wall is known as peptidoglycan. So this is the one term we can add, but obviously once you start reading this chapter, that is the moderns, or we can say cell bio, you will get to know a lot more things. So component is peptidoglycan. Peptidoglycan, or we can say it's nothing but the muramic acid is there, or we can say murine layer is there. So don't worry about that. After that, based on this cell wall, based on the composition of cell wall, the bacteria can be of two types. One is, we can say it's a gram positive bacteria, another one is the gram negative bacteria. So if you are talking about the gram positive bacteria, so you will make the bacteria colorful if it is a gram positive. So can you tell me what type of dye, or we can say what type of stain you will add to the gram positive bacteria? Safranin, or crystal violet? Safranin or crystal violet? Which type of stain you will use to dye or stain gram-positive bacteria? That's good. We say crystal violet, Brusil, good answer. Dragon, it's not safranin. Safranin is meant for the gram-negative bacteria. Okay. Good. Most of the answer I am getting like a mixed answer is there. So safranin as well as crystal violet. But the correct answer is crystal violet if it is gram positive. If it is a gram negative, then we can use the safranin. Okay. So here we'll go to the components. So components is peptidoglycan. Again, what is the function of cell wall? So can we say that the function of cell wall is again protection? Protection. Thereafter it's a providing the shape of bacteria. Providing the shape of bacteria. And based on the compositions, so gram positive and negative bacteria, gram positive and gram negative is having different composition, is having different compositions. So obviously these are the few things that we can so cell wall is dead in plants. They can't move like right. Yes, it's right. But obviously whoever is having the cell wall, so cell wall is the rigid structure. Can we say that this room is also like the room in which I'm teaching you, it is also surrounded by the cell wall, that is a wall? Or wherever you are there, or we can say at your place, can you see the cell? Can you see the wall of your room? Okay. So means you are having the wall in your room. So can you talk? Can you talk with the wall? So okay, few can talk. Obviously, it's having different type of power. So obviously, it's not. Why? Because the room is dead, or we can say the wall is dead. Same as it is here also. Here also, the wall is dead. So the main function of wall, why you are putting the wall in your room? So that no one can enter, or we can say if someone is known to you, only will enter inside your room, others won't. So these are nothing but the few things. So this is the cell wall. So can we say that this is the second layer of what? Second layer of cell envelope. And what's the third layer? What's the third layer? So can we say that third layer of cell envelope is nothing but the, so what's that? Tell me fast. So it's a cell membrane. So how many of you are having the cell membrane? Everyone or none? How many of you are having the cell membrane? Everyone or none? So obviously everyone is having, all is having the cell membrane. Everyone is having the cell membrane. If it is living cells, means the only thing 100% sure is cell membrane. The rest of the things I can't guarantee you, but I can guarantee if it is a living, then cell membrane will be there. Cell membrane will be there. So cell membrane is what type of a structure? So can we say that it's a living structure? If it is a living, can we say that it's a delicate, beautiful structure? So it's a living. So here we can say living. Thereafter, obviously it's not a thick structure. So here we can say it's a thin And the next one is, it's a delicate. Delicate means highly it's vulnerable. The moment you touch, it will change its shape. It will change its shape. So let's say this is my skin. So can I pull my skin? So obviously I can pull the skin. Why? Because the outermost layer is the cell membrane. Outermost layer is the cell membrane. But at the same time, can you do it with the plant cells? So can you do with the plant cells? Obviously, it's more than impossible. So you can't pull that, or we can say you can't simply change the shape of the plants. But in the animals, I can pull. I can change the shape. I can move from move from one place to another place. But at the same time, plant is not doing this. Plant is not doing this. So living, thin and delicate structure. And can we say it's a universal? It's a universal structure. If it is a universal, then we can directly say that it is present in all living beings. After that, what is the components? What is the components of this? So mainly it consists of protein and lipid. Mainly it consists of protein and lipid. So here we can say the components, or we can say it's a compositions. Compositions is mainly protein and lipid. But obviously protein is more in amount as compared to lipid. Protein and lipid. So after that, we'll move to the next part. So can we say that cell membrane surrounds the cytoplasm? Cell membrane surrounds the cytoplasm, or we can say enclose the fluid that is known as cytoplasm. So here we can say it surrounds the plasma of bacteria. It surrounds the plasma of bacteria. And next one is, it is so envelope is done. Envelope is done means envelope will start with what? The outermost one is glycocalyx. Just inside that you can find the cell wall, and just inside that the thin delicate structure that we were studying is nothing but it's a cell membrane. So mainly three structures are present in the cell envelope. So if someone is asking how many layers are present in the cell envelope of bacteria, so answer is it's 3. Answer is 3. But out of 3, how many of them are living? 1, 2, or 3? How many of them are living? So only one layer is living. That one layer is nothing but it's a cell membrane. While the first two layer, that is a glycocalyx and cell wall is dead layer, while the last one is it's nothing but the living layers. Okay, so is that clear? So first part we have done. So what is the first part? So first part is nothing but the cell envelope. After that, we'll try to enter into it, or we can say our topic is structure related to what? Structure related to cell membrane. So what are the structure that is related to the cell membrane? So I think in the last class we have we were discussing one structure. That one structure is somehow comparable to what? Comparable to mitochondria. Comparable to mitochondria. So comparable to mitochondria, can we call it as a mesosomes? And how the mesosome looks like. So can we say that mesosome is nothing but it's a membranous part, or we can say it's a part of cell membrane, but there is an infolding. There is an infolding inside that. Okay. So just I am drawing the part. So in the last class, I have drawn like this. I have drawn like this. So can we say that? Can we say that this infolding is nothing but the mesosomes? So here we can say it's a infolding. Achha, this infolding is inside or outside? Means towards the cytoplasm or away from the cytoplasm? So can we say that it is towards the cytoplasm? It is towards the cytoplasm. Okay. Generally, what is the color of bacteria? So bacteria, you can't say that bacteria is too colorful. Sometimes if it is having the pigments, that pigments will impart the color of bacteria. That color will impart the, or we can say pigments will impart the bacteria. So as such, we can't say that this is the color of bacteria. This is the color of bacteria. And obviously it is a microscopic in nature. So you can't say that this is the color. So how a mesosome work like a mitochondria? Why not anyone can do like the same thing? So here, mesosome is related with what? ATP production. And ATP, can we say it's an energy currency? Energy currency. So if you are having too much of ATP, what will you do? So obviously, you will be happy or sad after having a lot more ATP. So if you are having a lot more ATP, then even by walk, you can go to the Himalaya. By walk, you can go to the London, or we can say America. If you are having sufficient amount of ATP. If you don't have ATP, you are not even come out of your home if you don't have ATP. So that is the significance of what? Significance of ATP. Yes, everyone is happy, means everyone is having a lot more ATP, or we can say throughout the time they have synthesized a lot more ATP. So overall ATP is the most important thing. So here we can say infolding in the cytoplasm. After that, what else we can add? If there is an infolding, just imagine one straight line is there. One straight line is there. So if this straight line is having infolding, or we can say I have folded this straight line into the curve like this, zigzag pattern. So can we say that it is having the more surface area as compared to a straight line? As compared to straight line, this one is having more surface area. So here can we add one more point, that is increase in surface area. Next one is, can we have more points? So here we can say related to what? Related to respiration. So respiration, whenever you are having the food. So can we say that respiration is an oxidative process? Is an oxidative process where food is getting oxidized to give you a good amount of energy. to give you a good amount of energy. So that is the main function of respiration. And indirectly you are getting what? Indirectly you are getting a good amount of energy. So that is a source of food. So related to respiration. Next one is, and in your body, which part is doing this respiration, or we can say aerobic respiration? In your body, which part helps in the respiration, or we can say aerobic respiration? Lakshmi, no, not nose. Nose is somehow helping. Yes, Sadhana, that is a mitochondria. So mitochondria is a correct answer. Abhishek, it's a mitochondria. So overall, mitochondria is the correct answer. So mitochondria is the organelle at which the oxidation takes place. Oxidation takes place. And lungs, nose, they will just help you to fetch the oxygen, to bring the oxygen from where? To bring the oxygen from outside. Like let's say, I'm just inhaling. Inhaling means nose is helping, lungs is helping. So all the things are helping to get the oxygen from outside, nothing else. Nothing else. But at the cellular level, what organelles helps in the aerobic respiration? So we can say it's a mitochondria. Okay. Now we'll move to the next one is, so sometime it also helps in, or we can say, helps in what? Helps in synthesizing the cell wall. So here we can say it's a secrete cell wall. Next one is, if the question is coming, do the bacteria have DNA or not? Do they have DNA? The bacteria would have DNA or not? So answer is yes or no. Yes. Most of the answer I am getting. So answer should be, or we can say the correct answer should be yes. Correct answer should be yes. The bacteria is having DNA. And why DNA is important? Can anyone? I just want a good answer, or we can say it's like some innovative answer. Why DNA is required for the bacteria? What happens if bacteria lost its DNA? Lost its DNA. Obviously, what will be the main problem? Bacteria is not able to reproduce. Or once the bacteria started reproducing, so in that case, the offspring, or we can say progeny of bacteria will ask for the genetic material. Or we can say genetic material is nothing but a significance, or we can say it's your identity. It's your identity. Because here we can say the child of elephant is elephant or something else. So elephant's child will be elephant or tiger or some other part. So obviously I'm getting the answer elephant. So this chemistry, or we can say this story of elephant being elephant is hidden in what? Is hidden in the DNA. If the elephant is not passing its DNA to the next generation, so we can say next generation will not have the features of elephant. Okay, so that's why DNA is important for everyone. So what? Secrete the cell wall. After that we can go to the next one. We can go to the next one. Next one means it helps in making the copy of DNA. Or we can say this is known as DNA. It is nothing but the DNA replication. DNA replication. Yes, artificially we can do, but in artificial thing also DNA is required. DNA is required. If DNA is not there, means we can say software is totally gone. If software is not there, what is the use of hardware? So overall, this DNA is nothing but a software, or we can say it's a kind of language. Next one is, if DNA replication is there, means we can say it also helps in what? It also helps in cell division. It also helps in cell division. So these are the few parts that we have discussed in this mesosome part. So now my question is, mesosome is a part of cell membrane or cell wall? Mesosome is a part of cell membrane or cell wall? So obviously I will expect correct answer that, yeah, correct answer is cell membrane. Cell membrane. That's good. So mesosome is a part of cell membrane. Next one is, we'll move to the other one. So the first one is mesosomes. So second structure, what we talk is nothing but the one more structure is there, that is chromatophores. Achha, how many of you are autotrophic in nature? How many of you are autotrophic? So I think in this whole life, no one is autotrophic. So why you are not autotrophic? So can we say that you are lack of pigment system? You are lack of pigment system, and if pigment system is not there, you cannot harvest, you cannot harness, you cannot utilize the light. If you don't have the mechanism to utilize the light, means you are not able to synthesize your own food. You are not able to synthesize your own food. So that's why. But in some bacteria, I'm not talking about all the bacteria is having this. But some bacteria is having this chromatophore. If it is a chromatophore, means some pigments will be there, or we can say some pigment will be associated with this. So here we can say, yes, holozoic mode of nutrition. So here we can say associated with what? Associated with pigments. And if there is a pigment, means it can synthesize food. It can synthesize food. But this chromatophore is only present in autotrophs. And obviously not all autotrophs. Or we can say here, it's better to write here is, it's better to write present in autotrophs and mostly it's a cyanobacteria. Mostly it's a cyanobacteria. So yes, we are totally dependent on plant for what? Plant for food. Okay. So you want yourself as a autotrophs or heterotrophs? Being heterotrophs, you are happy, or autotrophs, you will be happy? Being autotroph, you are happy, or being heterotroph, you will be happy? So as a heterotrophs, you are happy. Okay, that's really good. Some answer I am getting. If I were I'll be autotrophs, I'll be happy. But most of the answer I am getting is, most of them are as a heterotrophs, they are happy. As a heterotrophs, they are happy. I don't know, but yes, it's good. Like you are just accepting your current situations. So that is still good. So it's a chromatophores. So after this, we'll move to the next. So here it's a chromatophores. After the chromatophores, few more structures that is related with the cell membrane is a fimbriae. So fimbriae is bristles like small bristles like structures. Small bristles like a structure. Bristles means small hairy like structures. And this hairy like structures helps in what? Attachment to the substrate, or we can say attachment to the substratum, or we can say attachment to the surface. So the first is helps in attachment. Helps in attachment to what? Attachment to surface. And surface, like sometimes it's a host surface, sometimes it's a rock, sometimes it's a stone, sometimes it's a soil, or many other things it can be. And here it's nothing but a small bristles like a structure. And one more important point is most of the pathogenic bacteria will have this bristles, or we can say this fimbriae. Most of the pathogenic bacteria. So here it's most of the pathogenic bacteria. Most of the pathogenic bacteria is having this. After that we'll move to the next one is. So 3 parts are done. What are the 3 parts? Fimbriae, chromatophores, and one more was there, that is the mesosomes. So here it's a. After that we'll move to the next 4th point that is also related with this cell membrane. So here we can say that is a pili. So pili, can we say that it is also the extension, or we can say outgrowth of what? Extension or outgrowth of plasma membrane. And it is somehow longer, or we can say somehow larger as compared to fimbriae. So fimbriae is chinna as compared to what? Pili. So pili is pedda as compared to this chinna part. So here it's a pili. Helps in what? Helps in the formation of conjugation tube. Helps in the formation of conjugation tube. Or if it is helping in formation of conjugation tube, means conjugation is the process in which this will help. So this is nothing but the fimbriae. After that, what to say? So here, next one is, after pili, it's a flagella. So 5th one is flagella, that is also related with the membranous. And what is the main usage of flagella? What is the main usage of flagella? Can we say it's a locomotion, or we can say movement? Movement is the main function of flagella. So it helps in, or we can say it is locomotory structure. It is locomotory structure. Okay. So these are the few parts. After that, the most important is its genetic material. The most important part is what is that? Genetic material. So what type of genetic material it is having? What type of genetic material? It is enclosed by the nuclear membrane, or the genetic material is just spreaded, just scattered in the cytoplasm. So can we say that it is spreaded, it is scattered, it is randomly present in the cytoplasm. It is randomly present in the cytoplasm. So after this naked, here we can say genetic material is nothing but the double stranded, but circular or linear. So here it's a double stranded circular DNA. But in this case, histone protein is present or absent? So histone protein is absent. But do you have histone protein? Do you have histone protein? Yes, your answer should be yes. You are having the histone protein, but the bacteria is not having the histone protein. So here we can say absence of histone protein. So what type of nucleus is there? It's a true nucleus or false nucleus? So we can say it's a false nucleus, and this is known as nucleoid, genophore, incipient nucleus. A lot more names we can give it to them. Okay, so here we can say a lot more name we can provide. So what are the name? We can call it as a nucleoid. We can also say that it's a genophore. We can also call it as a incipient nucleus. Incipient nucleus. So these are the term we can place it here for the genetic material. But genetic material is, or we can say DNA is only present here, or in some other part also genetic material is present, or we can say DNA is present. DNA is present. So can we say that one more structure, that is a plasmid, that is a unique structure which makes to look differently, which makes the bacteria looks differently from other bacteria, So the most important structure, that is nothing but the plasmid. That is nothing but the plasmid. So one more structure I am writing. Plasmid. Plasmid is most important for the bacteria. Most important for the bacteria. So first thing is, we can call it as a extra chromosomal DNA. Extra chromosomal DNA. Because normally DNA is present in chromosomes. But the scientists find some other structure which is also having the DNA. So that's why they can call it as a extra chromosomal DNA. This we can call it as a extra chromosomal DNA. And next one is one more part we can add. So what's that? So what's that part? In this case, it gives unique phenotype. It gives what? What type of phenotype? It gives unique phenotype. So does this structure replicate? Does this structure make its own copy? Yes, answer is yes, it can make its own copy. It can make its own copy. So here we can say it's a self-replicative. It's nothing but the self-replicative. So these are the few points we can add to the plasmid. We can add to the plasmid. So for today's class, will be up to this. So if you are having any doubt, then I will solve the doubt. Yes, time up. That is one doubt. Resistance to bacteria. Yes, that is good. Kirti, this plasmid is having different informations. So sometimes what happens, some person is infected with some other type of disease. So firstly, they will go to the doctor. Doctor will prescribe what? So doctor generally gives antibiotic. So once you have this antibiotic, but still your disease is persisting. Your disease is persisting means what? Means what? Bacteria is not showing any response to this antibiotic. Means bacteria is resistant to this. Bacteria is resistant to this. So that information is also present in where? In what? So this information is also present in the plasmid. Like if you are having penicillin, so penicillin, this plasmid, if this plasmid is having penicillin resistant gene, there is no effect of penicillin on this bacteria. Means that person will still have disease. That person is still having disease. Any other, what is histone? Histone is nothing but the protein, and protein is what? Amino acid. Amino acid. Main function of histone protein is to fold the DNA, into the properly compact structure. What gives protection to bacteria? Glycocalyx or cell wall. See, combinedly, glycocalyx, cell wall, and cell membrane, all are providing the protection. All are providing the protections. Bacteria is resistant to certain antibiotics. Have to increase the doses or change. Either you can increase the doses, but still you are not cured, then you can change, or we can say shift to some other. So I am just solving the doubt, nothing else. Okay, so bye bye everyone. We'll meet you in the next class. Till this time, take care of yourself. Have your breakfast if you didn't have.